Hey guys, welcome to 11.2. We are continuing our discussion on cellular communication. So if you remember last time we talked about transmembrane proteins, and so now we're going to be talking about how molecules are going to bind to transmembrane proteins, which make it change its conformation or make it change its shape. And this is going to allow for the message to be sent inside. And so there are three types of cellular responses that we're going to talk about today. The G protein coupled receptor, we're going to call these GPCRs because it's just fewer syllables, and receptor tyrosine kinases. And so in here you should see a couple words jumping out at you, tyrosine and kinases. These should sound familiar at this point. And then ion channel receptors. These should also sound familiar from when we were talking about nerves and nerve cells and how when molecules pass from the postsynaptic cell to the presynaptic cell to the postsynaptic cell, the neurotransmitters are going to be interacting with these ion channel receptors. So this should all be a little familiar, bits and pieces. So let's first talk about the G protein coupled receptors. This is going to be the more intense of the three signaling process that we're going to talk about, so let's get this one done. There's a few steps, and I recommend creating a drawing, a labeled drawing, to wrap your head around this after you go through the video and after you do your reading. So a G protein coupled receptor, the GPCR, is going to be the transmembrane protein, and this is what's going to allow for the message to be brought from the outside to the inside, because we're going to have something to bind at the binding site. It's going to change the conformation. And so here, if we look at this, here's our GPCR, and it's going to be coupled with a G protein. And here's our G protein. And this G protein is going to be sort of lodged in that membrane near the GPCR. And there's going to be, know that at this point, that there's an active and there's an inactive form of this G protein. And so when it's in use, it's obviously activated, and when it's not being used, it's inactivated. And so these are going to work together with usually an enzyme. And so here is our enzyme. And so here, what's going to happen is that this G protein, or when there is a message and the GPCR is going to change its conformation, it's going to allow this G protein to move towards it. And then it's going to move back towards the enzyme. So this G protein is lodged in the membrane, but it moves back and forth between these two, between the GPCR and the enzyme. So when a signaling molecule attaches to the external side, so here it is, our signaling molecule, what's going to happen is that this, like we said, this G protein is going to attach onto, that's going to bind to the GPCR, and it's going to get rid of a GDP. Remember we talked about GDP, guanosine diphosphate, so that's our inactivated version. And remember that the triphosphate is going to be our activated version. And so we're not talking about energy here, we're talking about a signaling molecule, even though those molecules are very similar. So when the G protein attaches onto the GPCR, it's going to get rid of the old stuff, the GDP, the inactive, and it's going to make room for a fresh new GTP, which is going to activate it. So this GTP is going to activate it. Then, when it's activated, this G protein is going to move towards the enzyme, and it's going to cause a conformation in the activated enzyme, or in this enzyme. Once this changes conformation, it's going to allow for this to cause a cellular response or for a transduction pathway within the cell. So just know that there's more steps later, that this is the initiation, that this is going to initiate steps or a cellular response. And so here it is going to have the GTP, it's going to be activated. Know that this ligand here, or this signaling molecule, is going to continuously attach and detach. It's not used just once, it's not one and done. So the more signaling molecule that you have, the more times you're going to send this message to the activated enzyme or to create a cellular response. So if there's lots of ligands here, if there's lots of signaling molecules, lots of hormones, they're all going to be attaching and reattaching, causing a greater response. The fewer that are there, then it's going to be causing less of a response. And so here, note that the enzyme also functions as a GTPase. And so this should make a lot of sense. What it's going to do is it's going to cut one of those phosphates off. It's going to get rid of it. And instead of releasing energy, what it's doing is it's inactivating this GDP. So remember that the GDP is the inactive molecule, whereas the GTP is the activated. So what this enzyme does is it deactivates it. And this is important because what that allows is for the signal pathway to stop when there is no more, very quickly, when there is no more signaling, when there are no more signaling molecules out here. So once there are no more signaling molecules, once there is no more external message, this pathway is going to be able to turn off very quickly every single time, so that the message isn't continuously being sent when there is no message to be sent. So GPCRs are extremely widespread and diverse in their functions. They are going to be used very often in embryonic development because there's lots of communication in embryonic development. There's lots of cells that are growing and developing, and this is also used in sensory reception, so smell, taste, vision. And so know that if we're depending on something, if something is very important in our cells, when they malfunction, that's going to cause diseases. Remember that we think of diseases as symptoms, what we see in this macro world. But in reality, diseases are something that's going wrong within our cells. And so malfunctions in our GPCR are involved with many diseases, bacterial infections like cholera or pertussis, which is a whooping cough, are just a couple of them. Second cellular response we're going to talk about are RTKs, receptor tyrosine kinases. And so these, once again, are transmembrane proteins that are going to have ligands or signaling molecules that are going to attach externally. And at the inside, the internal part of the transmembrane proteins are going to have tyrosines on the inside, multiple. And so this should remind you that phosphates can bind to tyrosines. So this should indicate a little bit as to what's going to happen. So here you can see that they span from the outside to the inside, and it is drawn a little bit differently here, but they still have the alpha helices. It's just a different type of drawing. And so let's take a peek as to what happens. So the binding site of the signaling protein, an example of this would be like growth factor, causes, so here we have a signaling molecule, and they're going to attach. And what this causes is for both of those transmembrane proteins to associate or to grow closer to one another. And this is called dimerization. Because they get a little bit closer to one another, they're going to, this can happen with just two, or it can be in large clusters, which would cause a greater signal. But this is going to sort of activate these molecules. Once they come together, their closeness is going to activate these tyrosines, or these molecules. And when dimerization occurs, these tyrosines are then going to add phosphate groups to each one of these tyrosines. And so this is an activated or a fully activated receptor tyrosine kinase. And it is ready to go. It is ready to send a message because it has all of those phosphates on the tails. So now that the receptor is fully activated, once this happens, once those phosphates are on there, it's going to trigger a response. These proteins are going to be, they're going to attach to these RTKs, and it's going to change their conformation. So only when the phosphates are fully loaded, those proteins, those relay proteins, are going to all of a sudden recognize it and be able to attach. And then it's going to change their conformation, and that's going to create a transduction pathway that's going to send a response to other proteins or other molecules, which is going to eventually lead to a cellular response. And so why would a cell develop more than one way to signal, to send a signal externally to internally? Well, the GPCR is going to activate just one signal transduction pathway. However, this is going to be a broader tool. So one RTK molecule here can activate more than 10 different transduction pathways. It's going to be a greater tool, or a tool that's going to allow for a larger message to be sent. And so this is going to allow a message to be sent more quickly. And just like we said before, that diseases occur in cells. So here, if this RTK is functioning or is malfunctioning, oftentimes these are associated with types of cancer. So if we think this won't turn off, let's say this stays in an on state, and it's sending a message to the cell to very kind of oversimplified, but to divide, continue to divide, and it doesn't turn off, that cell is going to continue to divide, and then you're going to have uncontrolled cell growth, which is what cancer is. So those are RTKs. Our third cellular process for sending messages externally to internally that use transmembrane proteins is ion channel receptors, and these should sound familiar. We talked about them. We talked about nerves or nerve cells. So what happens is the ligand or the signaling molecule is going to attach to the protein, and it's going to open the gate, and it's going to allow for a rapid change in the concentration of a molecule, of an ion, on the inside. So it's going to allow ions to travel quickly from externally to internally, or vice versa. And so when the ligand or the signaling molecule is attached to the protein, then it's going to open, and when it detaches, it's going to close. So that is our third process, but we have one more intracellular receptor, or one more process we're going to talk about, and this is intracellular receptors. So this is our receptor. Before we were talking about transmembrane proteins, right? Because we had to take the message from outside of the cell and bring it in. But if we're talking about intracellular receptors, we're talking about hormones or signaling molecules that are going to be small and nonpolar. This is going to allow them to travel through the membrane, and so that they can interact with molecules on the inside. And so when a hormone is going to travel through the lipid membrane, it's going to attach onto a receptor complex and become a hormone receptor complex right here. Or it's going to attach to a receptor and become a hormone receptor complex. An example of this we can remember when we were talking about our kidneys. When the aldosterone travels through the lipid membrane, attaches onto the receptor, creating a hormone receptor complex. That's going to send a message, which is going to turn things on and turn things off in the genetic material, and it's going to allow for the blood volume to be altered. And so those are all of the different processes that we're going to talk about today. I do recommend drawing some of these out to help you remember them. Have a great day.